If you work with cells, you know one of the biggest challenges is keeping good viable stocks. One of the best ways to do this is through cryopreservation, the process of freezing cells so they can be thawed and used at a later time. Cryopreservation is a method of preserving biological tissue or cells at ultra-low temperature. At such temperature, metabolism gets virtually halted. Storing cultured cells in liquid nitrogen with complete medium in the presence of a cryoprotective agent such as dimethyl sulfoxide, DMSO, or glycerol is the best method for cryopreserving cultured cells. When freezing cells, several crucial factors must be considered to ensure their viability and integrity upon thawing. Firstly, it's essential to freeze your cultured cells at a high concentration and at as low a passage number as possible. This ensures the maximum survival rate and functionality of the cells. Make sure that the cells are at least 90% viable before freezing to reduce the risk of losing valuable cell stocks. Using suitable freezing containers, such as cryovials or single-use bioprocessing containers, is vital for storing cells in liquid nitrogen for long-term preservation. These containers are designed to withstand ultra-low temperatures and prevent contamination. Selecting the appropriate cryoprotectant, such as DMSO or glycerol, based on the cell type, is another critical factor. Determining the optimal cell density and volume for aliquoting is also essential. Too high or too low a cell density can affect the freezing and thawing process, leading to cell damage or loss. Finally, defining the freezing method to best control the cooling rate is crucial to prevent cell damage during freezing. Whether using a controlled rate freezer for slow freezing or immersing cells in liquid nitrogen for rapid freezing, the goal is to minimize the formation of ice crystals that can damage cell structures. Considering these factors will help ensure the successful cryopreservation of your cultured cells, maintaining their viability and functionality for future use. Freezing adherent cells or cells in suspension presents distinct challenges in the cryopreservation process. Adherent cells, which are attached to a substrate, require additional steps for successful freezing. Prior to freezing, these cells must be detached using enzymes like trypsin or EDTA. Once detached, the next step is to resuspend the cells in a suitable freezing medium containing cryoprotectants. On the other hand, cells that are already suspended in culture media can be directly mixed with the freezing medium for preservation. This straightforward approach simplifies the process, making it easier to achieve uniform cryopreservation. Freezing cells is a multi-step process. Each step is crucial for ensuring the viability of the cells after thawing. The first step is preparation. This involves harvesting the cells and preparing them for freezing. The cells are typically suspended in a freezing medium that helps protect them during freezing. The next step is the actual freezing process. Freezing is typically done using a controlled rate freezer. This device gradually lowers the temperature approximately 1 degree C per minute. Alternatively, place the cryovials containing the cells in an isopropanol chamber and store them at 80 degrees Celsius overnight. Transfer frozen cells to liquid nitrogen. If you have any questions about cryopreserving your cultured cells, please contact us.